The overall goal of this procedure is to assess myofibril and cardiomyocyte maturation in the developing embryonic mouse heart. This is accomplished by first orienting and freezing the embryonic heart in cutting medium. Next, the heart is cryosectioned in the proper orientation. Then the heart sections undergo immunofluorescent labeling of proteins of interest. Finally, confocal microscopy of immunostained heart sections is carried out. Ultimately, two-dimensional and three-dimensional image analyses are used to show the development of myofibrils and other cardiomyocyte structures.
This method can help answer key questions in the cardiac development field, such as how mutations in cardiac genes affect myofibril assembly and the emergence of specific structures, such as intercalated discs and costomeres.
To snap freeze embryonic hearts, use optimal cutting temperature or OCT medium to fill a 3.5 cm petri dish. In a chemical hood, cool 2-methylbutane in liquid nitrogen. After isolating mouse embryonic hearts according to the text protocol, place the hearts in OCT and allow them to equilibrate for several seconds before transferring them to a 7 mm mold containing OCT. Orient the interior wall of the heart to the bottom of the mold. Gently place the mold into the liquid nitrogen-cooled 2-methylbutane, taking care not to allow the 2-methylbutane liquid to touch the OCT or heart. Freeze until the OCT is solid white, then transfer the mold to an ice bucket containing dry ice. After all the hearts are frozen, wrap the cryomolds in foil and store at -80 degrees Celsius until ready for cryosectioning.